how they will work. What is the exact mechanism? See, this is your cell. This is the nucleus. Nucleus are having DNA. DNA will make RNA and all those things. And this is your beautiful cytoplasmic receptor. Now, when ligand enter inside the cell, the first step is what? Ligand enters the cell and binds the receptor in the cytoplasm. So ligand will bind with the receptor. The first step is ligand will bind with the receptor. Okay. The receptor is initially bind to heat shock protein. Which protein? Heat shock protein, also called as chaperone protein. This is a very important topic I'm going to tell you. This is a topic which is coming again and again, and we are tend to make these mistakes. So even before ligand come inside the cell, cytoplasmic receptor are binding to heat shock protein. Why? To make them inactive. Why? To make them inactive. What is the answer? Why you need chaperone protein? Why you need heat shock protein? To make them inactive. Otherwise, they will enter in the nucleus, activate the DNA without the ligand. So without the ligand, your receptor need not be activated, my dear doctor. It need to be activated only in the presence of hormone. The receptor is initially bound to heat shock protein to make them inactive. When ligand binding will happen, it will cause dissociation of heat shock protein. So it will dissociate the friendship with the chaperone and it will form homodimer. It will dimerize. It will form homodimer. The third step is what? Dimerization of the receptor. Dimerization. Hetero or homo? Homodimer. After dimerization, this will translocate to the nucleus. Now this will translocate to the nucleus and bind to the HRE. Bind to the hormone response element on the DNA and then regulate gene transcription. Why I'm telling you this detail? Because this is a topic which is often repeated for NEET PG and INICET. So I hope you can recap now. There are two types of intracellular receptor, type 1 and type 2. This much is clear. Type 1, they have ligand of all the steroid hormone. It can be aldosterone, estrogen, testosterone, and all those things. Type 1 hormones are receptors are also called as homodimer. What are the steps? So ligand enters the cell. Step number 1, ligand enters the cells and binds the receptor in the cytoplasm. The first step. Number 2, it will remove heat shock protein, which is also called as chaperone protein. Chaperone protein. Next MCQ. What is the need of chaperone protein? What is the need of heat shock protein? The need of heat shock protein is that to make the receptor inactive. Otherwise, even in the absence of hormone, it will go inside the nucleus and activate the DNA, RNA, and protein. It should not happen. Only when ligand in the form of hormone enter inside the cell, it will dissociate the chaperone, forms dimer, translocate in the nucleus, enter and bind with the hormone response element and regulate gene transcription. So this is the exact mechanism given in Harper and Ganong, which is asked again and again in INICET NEET PG. I hope you should not forget. There should not be any doubt. It is a common question for physio, biochemistry, and medicine.